Today, we'll discuss about the control of microorganism. So, prevention, control, or elimination of microbes that are potentially harmful to humans is one of the primary goals of our healthcare system. The process by which all living cells, viable spores, viruses, and viruids are either destroyed or removed from an object or habitat is termed as sterilization. A microbial population is not killed instantly when it is exposed to a lethal agent. Like population growth that we have discussed earlier, population death is generally exponential or logarithmic. That is, the population will be reduced by the same fraction at constant interval. So when the population has been greatly reduced, the rate of killing may slow due to the survival of more resistant strains of microorganism. So there are certain approaches for controlling microbial growth, and the most commonly used are the physical and the chemical methods. First, we'll see the use of physical method in microbial control. In physical methods, the most commonly employed physical agents are heat, filtration, and radiation. So, heat. Heat is the most widely used lethal agent for sterilization. Objects may be sterilized by dry heat in an oven or by moist heat, which is provided by steam. Of the two methods, sterilization by dry heat, it requires a much greater duration and intensity because heat conduction is less rapid in dry than in moist air. In addition, bacteria can survive in a completely desiccated state, and in this state, the intrinsic heat resistance of vegetative bacterial cells is greatly increased, almost to the level characteristic of spores. Consequently, the death rate is much lower for dry cells than for fully hydrated ones. So this dry heat of pressure. At this temperature, saturated steam, it destroys all the vegetative cells and endospores, and this treatment is continued for about 15 minutes to provide a margin of safety. And another method of heat sterilization, which we commonly call it as pasteurization, was developed by Louis Pasteur. Pasteurization involves heating a particular food, such as milk, to a specific temperature long enough to kill the spoilage microorganism. Many different time and temperature combinations can be used for pasteurization. The long temperature, long time process involves bringing the milk to a temperature of 63 degrees Celsius for 30 minutes, while the high temperature short time, also known as flask pasteurization method, brings the milk to a temperature of 72 degrees Celsius for 15 seconds to kill the spoilage microbes. Next, we'll come to filtration. So an excellent way to reduce the microbial population in the solution of heat-sensitive material is filtration. So there are two types of filters. The first one is the depth filter and another one is the membrane filter. Depth filter, it consists of a fibrous or granular material that have been bonded into a thick layer which is filled with twisting channels of small diameter. The solution containing microorganism is sucked through this layer under vacuum, and the microbial cells are removed by physical screening or entrapment, and also by adsorption to the surface of the filter material. Depth filters are made of diatomaceous earth, unglazed porcelain, asbestos, etc. While membrane filters are circular porous filter with little over 0.1 millimeter thick and the pore size of about 0.2 micrometer in diameter, and these are made up of cellulose acetate, cellulose nitrate, polycarbonate or other synthetic material. The membranes are held in a special holders and often preceded by depth filter to remove larger particles that might clog the membrane filter. Further, the solution is pulled or forced through the filter with a vacuum and collected in the previously sterilized container. So another type of filtration is the air filtration, and most common example is the surgical marks and the cotton plugs on culture vessels that let the air in but keeps the microorganisms out. While laminar airflow biological safety cabinets employing high efficiency particulate air filter which remove 99.97% of 0.3 micrometer particle are one of the most effective air filtration system. These cabinet forced air through the HEPA filter, then project a vertical curtain of sterile air across the cabinet opening. They are also used in research lab and industries where a sterile working surface is needed. Next, we'll see radiation. So this radiation in various forms, from high energy radiation to sunlight, can be used to kill microbes or inhibit their growth. Generally, there are two types of radiation used in sterilization, that is ionizing and the non-ionizing radiation. Ionizing radiation is an excellent sterilizing agent, and this can penetrate deep into the objects. They include the X-rays and the gamma rays, and these are strong enough to pass through the cell where it alters the molecular structures and damages the cell components. They introduce break in the double-stranded DNA, which causes mutation to occur, and ultimately, when the mutation accumulates, it leads to the death of the cell. While the most common form of non-ionizing radiation is the UV radiation. They effectively reduce the microbial population where direct exposure takes place. UV rays, it exerts its mutagenic effect by exciting electrons in the molecules. These excitation of electrons in the DNA molecule results in the formation of extra bonds between adjacent pyrimidines, which is specifically the thymines in the DNA. The bonding of the two thymines is called the thymine dimers. These dimers, it change the shape of the DNA in the cell and it causes problems during replication and ultimately it kills the cells. Now we'll come to the use of chemical agent in microbial control. Chemical agents reduces the microbial population, but it generally not intended to achieve sterilization. Chemicals are more often employed in disinfection and antisepsis. Here, disinfection is the elimination of microbes from the inanimate objects or surfaces, while the removal of microbes from the living tissues are termed as antisepsis. So an ideal disinfectant or antiseptic must be effective against a wide range of infectious agents at high dilution and in the presence of organic matter. Although the chemical must be toxic for infectious agent, it should not be toxic to people or corrosive for common materials. It should also be stable on storage, soluble in water and lipids for penetration into microorganism, and have a low surface tension so that it can enter cracks in the surfaces. A large number of chemicals are in common use, which include the first one is phenol and phenol derivatives. Phenol was the first widely used chemical as a disinfectant and antiseptic. Phenol and phenolics such as cresols, gylinols and orthophenylphenols are used as disinfectants in laboratories and hospitals. Phenolics, it damages the cell membrane and denatures the proteins of the microorganism. They are also effective in the presence of organic material and remain active on surfaces long after application. Next is alcohol. Alcohol is also one of the most effective and widely used antiseptic and disinfectant. They are bactericidal and fungicidal but are not effective on bacterial spores. Ethanol and isopropanol are among the most popular alcohol germicides and are usually used in the concentration of 70 to 80%. The mode of action of alcohol is that it solubilizes the lipid bilayer of the cell wall and the cell membranes, and it creates a pore which further enters into the cell cytoplasm and denature the cellular components, which ultimately kills the bacteria. As alcohols evaporate very quickly, they leave no residues and are useful in degerming the skin before injections. Further, for disinfecting thermometers and small equipments, a 10 to 15 minutes soaking in alcohol is sufficient. Next is halogen. Among the halogens, iodine and chlorine are used as antimicrobial agent. Iodine is used as the tincture of iodine containing a 2% solution of iodine and sodium iodide in water ethanol solution. They are commonly used as a skin antiseptic, but it may damage the skin and the stain is left, which result in iodine allergies. A combination of iodine and organic molecule called the iodophore is developed, which are less irritating than iodine and do not stain. So these iodophore are water-soluble, stable and non-staining and release iodine slowly to minimize skin burns and irritation. Iodine are bactericidal, fungicidal, virucidal and even sporicidal. Chlorine is widely used to disinfect municipal water supplies, swimming pools, and they are also employed in dairy and food industry. They are used as the sodium hypochlorite and calcium hypochlorite. And the mode of action of chlorine is that it carries out oxidation of the cellular materials and destruction of vegetative cells of bacteria and fungi, but not spores. Now we'll come to heavy metals. Heavy metals such as mercury, silver, arsenic, zinc, and copper are used as antimicrobial agent. Heavy metals and their compounds inactivate cellular proteins and enzymes by combining with them. When they are present in higher concentration, heavy metal salts, it coagulates and precipitate cellular proteins and kills the microorganisms. Next is aldehyde. Among the aldehydes, formaldehydes and glutaraldehyde are commonly used antimicrobials and are highly reactive molecules that combine with cellular proteins and it inactivates them. They are sporicidal also. So this formaldehyde before use is usually dissolved in water. They are stable in higher concentration and at higher temperature because in room temperature they are polymerized to form a para-formaldehyde. Another aldehyde, that is glutaraldehyde, is used in 2% buffered solution as a disinfectant. It is less irritating than formaldehyde and it is used to disinfect hospitals and laboratory equipments. Glutaraldehyde usually kills the vegetative cells within 10 minutes of exposure but requires about 12 hours to destroy the spores. Next is gaseous agent. Ethylene oxide and the beta-propyl-lactone are the commonly used gaseous sterilizing agent. Heat-sensitive items, these are sterilized by the use of ethylene oxide. They are both microbicidal and sporicidal. And it is one of the most effective sterilizing agent because it can rapidly penetrate packing materials and plastic wraps. Extensive aeration of the sterilized material is necessary to remove residual ethylene oxide as it is very toxic. While beta-propyl-lactone is occasionally used as sterilizing gas, they kill microbes more rapidly than the ethylene oxide, but they cannot penetrate materials well and is carcinogenic, and hence they are not used extensively as ethylene oxide. So that was about the control of microorganisms by physical and the chemical agent. And in the end, although many microorganisms are beneficial and is necessary for human well-beings, microbial activities may have undesirable consequences such as food spoilage and diseases. Therefore, it is very much essential to kill the microbes or inhibit their growth to minimize their destructive effects. So when working with microbes, operations that might generate infectious aerosols, it should be carried out in biological safety cabinets. Benchtop and incubators should be disinfected regularly, An autoclave must be maintained and operated properly to ensure adequate sterilization.